Hi, hello. This is the topic number 20, the muscles, and finally we'll talk about the muscles. Let's start with the and I think first is that how the muscle is regulated by the calling. This better. Yes, how the muscle is is controlled by the nerves. So first of all, um, muscle control. First of all the skeletal muscles skeletal muscles are controlled by somatic motor neurons, which means it is controlled by our say control voluntarily controlled, and say this is a muscle. I'm trying to draw something that a muscle have multiple muscle fibers, and then what is a motor, and then each muscle, this may be somewhere far away. This is a motor neuron, and then it control this one and this one and probably this one. And so this one, two, three muscle fibers plus this motor neuron, we call them the motor units. Why we call it a motor unit? Once each muscle fiber is stimulated, all myofibrils will contract together, and the whole muscle fiber is on and off pattern. And then once one motor neuron is activated, all muscle fibers connect to one motor is all connected also. So it's all activated also. So a motor neuron means a motor neuron has one neuron and multiple muscle fiber. And multiple muscle fiber, all of those are from the same muscle. However, let's say this, the same muscle has multiple muscle fibers. Not all of those are controlled by this same neuron. This one. So this muscle fiber controlled by another motor neuron. So in this case, is how the later at the end of this topic will say how the muscle strength is regulated by regulate how many motor units is being activated. The more motor units are activated, the more muscle strength it will be generated. This is a motor neuron, and then next let's talk about the neural muscular junction. What is a neuromuscular junction? Oh, I hate my handwriting. Why so bad? Whatever. Neuromuscular junction is basically it's a synapse. So, and this is especially a synapse between a motor neuron and a muscle fiber. This muscle fiber, in most cases, this neuron, this NMJ, neuromuscular junction, is in the center of the muscle fiber, and this is the end. This is the axon terminal end. And this has many things in special. One thing is capsulized. This has a capsule. This capsule is and it completely enclosed the whole cleft. So in this case, and then it release things called acetylcholine. Acetylcholine is the neurotransmitter between the motor neuron and the muscle fiber. However, ACH is not special because ACH is used by many things else. And the special thing is that the enclosed clefts. So in this way, the ACH concentration can be very, very high. And also this motor neuron normally is spread out. The terminal end spread out into multiple branches, and each multiple branches falls in this called NMJ. And this again, this is encapsulated, and there's a I forgot the name of the cells. You can double check what's the name of the cells. Cells completely enclose this one, and also at the end of those. So this is the motor presynaptic terminal, and then inside the muscle here, the muscle also enfold. This is called what's the name of this? Some junction, folded junction. Oh, I forgot the name of it. And then this special area on the muscle side, this is called end plate. End plate is also specialized by enfolding the memory, something like this. And this enfolding, on the top of it, there's a receptor of ACH or ACHR is a receptor. And by enfolding of those membrane, there's a lot of ACHR located on the end plate. And in these cases, if you remember a motor neuron, a regular neuron here, this is the dendrites axon, and this is HELOC. You need a summation to accumulate enough electric potential here to activate action potential, and this action potential summation needs to be activated by multiple stimulation here from the dendrites. But from here, in the case of NMR, one action is enough to promote action potential, all because of the enfolding and high. So firstly, high concentration of ACH, and then large amount of ACHR to trigger an action potential. Now you have an action potential here. And further on, this action potential need to be very short because you don't that's for the purpose of a very accurate control from the timing purpose. And at the same time, so there's another on the surface of here, there's another enzyme called ACHE enzyme, which can very fastly degrade ACH in acetylcholine to acetyl and choline. And this is the major structure function of the NMR neurotransmitter neuromuscular junction. And let's see what happened next. What happened next is that in the muscle fiber, there's a large enfolding. This is end plate, and this ACHR here. And when the action potential is generated here, it go all the way here, going deep in a regular neurons. The action potential will propagate through cell membrane, and say this is motor neuron. It want to generate is propagate through the axon. In the case of muscle, it will propagate inside the muscle because enfolding. This enfolding called a transverse tube or tubule, or just T-tubule, transverse tubule. Because of transverse tubule, this action potential can be delivered inside. And if you have a large muscle fiber, you see transtubule tubule is all around the cell membrane. And this cell, by the way, the cell membrane of the muscle fiber is called sarcolemma. In muscle fiber, many special cell structures has a special name because muscle fiber cells is it's a long cell, it's not special cell. And then when it going inward, let's use some special color here. There's a special protein here. Oh, you call it. I need to double check. Right? Yes, DHPR. Yes, this protein called it DHPR, and this is a voltage gated calcium channel. When it's open, it allows the calcium to get in. However, when the muscle contraction, you don't really have a lot of calcium outside here. So even though the DHPR opened, not much calcium to get in. What's really make it happen, making the muscle contract is the another thing here. We call it here. The black thing I'm drawing this one. This is the sarcoplasmic reticulum. The sarcoplasmic reticulum contains a lot of calcium. So basically, the muscle fiber carries its own calcium as a reservoir of calcium. In normal cases, when the muscle is not contracted, all calcium stay in the reservoir, and this reservoir has a turn on and off. This green one, on and off cap. This is called RYR. RYR is very important protein because RYR is very close to DHPR. When DHPR change shape, RYR also change shape, and then release the calcium outside the sarcoplasmic reticulum. And what does calcium do though? Last time we mentioned that calcium will remove the troponin, remove the troponin from the actin, F-actin, and then the F-actin, then myosin will bind to F-actin, the muscle will contract. This is how the muscle is contract. And the majority trigger of the muscle contraction, the calcium coming from the reservoir, the sarcoplasmic reticulum, there's reservoir calcium. And then let's next talk about, I think it is the I believe in my slides was next in my slides. Yeah, energy supply. So this step use energy, and all energy we have from ATP. So all of those muscle contraction, the direct energy came from ATP. And then what ATP going to do? 60% ATP going to the myosin contraction. And myosin drag the actin F-actins bind inward, and then when they return to the poised status, they need ATP to go back. So it's 60% to go to a myosin, and then 30% of those is coming from something else. So let's draw this again. This is T tubule, and this is the sarcoplasmic reticulum, and then this is a DHPR, and this is RYR. When RYR is open, calcium. When this calcium, calcium going out, and then the calcium, one calcium going out is only temporarily regulating muscle contraction. Is for all of those muscle contraction, timely regulation is extremely important. So for that reason, the calcium once get out, they need to get it back. How they get back? Here, this is a calcium pump. to pump calcium back to the sarcoplasmic reticulum. And in this case, in the muscle contraction, 30% of the calcium, 30% ATP is for the calcium pump. And then we have about 10% left. What's the 10%? Here. You have action potential here. When you have a generate action potential, you always have to have the resting potential, and the muscle resting potential is minus 80, I think, or 90. And then to maintain this resting potential, you obviously have sodium out and potassium in, and then 10% of energy coming from the 10% of ATP is used to maintain the resting potential. And then how much do the muscle have? A lot of ATP? A lot ATP? No, it's not a lot. One muscle fiber contain ATP only lasts about 1 second. And when the muscle store ATP is used up, some other thing called creatine phosphate or phosphocreatine or phosphocreatine convert into ATP. So they provide some immediate supply of ATP. And when your muscle is under resting, you only need this, only about 10%. And then this gradually is maintained by, let me see. And this energy is mainly maintained by fatty acid. So when the muscle is under resting status, most of the energy came from fatty acid. And when the muscle start moving around, and when it first use ATP for sure, and then this ATP only lasts 1 second. And after that, creatine phosphate convert to ATP, and this creatine phosphate lasts for only about 10 seconds. So next, when the muscle activate, the major energy resource is glycogen. Again, when the muscle is resting, fatty acid. When the muscle is active, glycogen. And at the beginning, glycogen mainly go through the aerobic respiration. Glycogen become glucose, and glucose becomes CO2 and water and ATP. One glucose have 36 ATP generated. In the mitochondria, the muscle have a lot of mitochondria, and so this is muscle, and you say, you have muscle fibers here, muscle fiber. Let's see, not really. It should be muscle itself should be, say, not that color. Muscle itself should be black, and then you have something, a lot of mitochondria stay in the muscle, so they can produce ATP from the glycolysis of glucose. Oxidation of the glucose. And in this status, you also need a lot of oxygen. And for all of by the way, the muscle fiber, skeletal muscle, has a very high content of mitochondria, but it's not the highest content. Probably the second high content of mitochondria. So which tissue has the B highest mitochondrial content. That's the heart muscle. We talk about in the next semester. And also in the muscle, you see, you need meat. Meat most eat the muscles. Some of those are red. Why they're red? Partially come from the mitochondria because mitochondrial cell has color, and the majority of those red color came from something else. This red color is similar to similar to the blood red color, and this is very similar. This thing called myoglobin. In the blood, we have hemoglobin. Now here, we have myoglobin. Both hemoglobin and myoglobin have one function: carry oxygen. And muscle is both heart muscle and skeletal muscle, and probably some of those, and also some of those smooth muscles, are the only organ that can store oxygen. No other, I believe no other organs can store oxygen here. And so the muscle store their own oxygen. All of those just make sure also the muscle can have a large, fast and high capacity of ATP production from the glucose, even though that the muscle have so many properties to make sure they produce a lot of oxygens. There's still sometimes we do not have enough oxygen to fully oxidize glucose into carbon dioxide and water and generate 36 ATP. In that cases, the muscle have to use the anaerobic glycolysis. What is anaerobic glycolysis? This is one glucose become 2 lactate plus 2 ATP. You see, it generates way less ATP than the complete oxidation. However, it's much faster. It does not require oxygen. That's the key thing. It does not require oxygen. And at the same time, then you have a lot of lactate to be accumulated in the muscle. And what happened to those lactate? This is a long-term misunderstanding. In a very early study, they believed accumulated lactate accumulated lactate is the cause of muscle fatigue. One thing for sure is that accumulated lactate does cause muscle fatigue. However, there's no accumulated lactate because lactate very fastly get into the blood, and then when they get into the blood, some of those will be excreted by sweating through the skin, and some of those will go to the liver, and to be convert back to glucose in the liver, and some of those will be consumed by the kidney. Either way, overall, if you have an increased lactate, it's mainly a systemic increased lactate, and the body does not allow that. It will have many different ways to get rid of the lactate. The lactate does not accumulate in the muscle. Why people have this understanding at the very beginning? That's because at the very beginning they use the frog leg muscle to study, and the frog leg muscle is isolated. It's insulated muscle without blood supply. That's why you have accumulated lactate. And then I think we're almost done. And so what else in mind? What else in mind? Not here. Oh, yes, I'm almost done. So, yes. Yeah, a muscle based on how much mitochondria and myoglobin they have. There's mainly two types type 1 muscle fiber, also called fast twitch, and type 2, also called it slow twitch. I need to double check. I need to make sure I'm right. Fast and oh yeah, I was wrong. So fast one is slow because it has high mitochondria and high myoglobin, and type 2 is fast. It has low mitochondria, and in these cases, type 2 is mainly for fast response, and type 1 for increased endurance. And I think that's and also there's and I need to get a new one. It's common sense muscle can get tired. The process of muscle get tired called muscle fatigue. And what causes the muscle fatigue? Again, at the very beginning, people believe it's a lactate accumulation. This is wrong. It's not by lactate accumulation, as mentioned. Lactate does not accumulate in the muscle in the living body. So, and then multiple hypotheses has been proposed. One of those is that given time, long in time, it's called central fatigue. Given long in time, the central nervous system cannot generate enough neurotransmitter, and without enough neurotransmitter, muscle cannot contract. And this cannot fully explain everything. It seems like only when the body is in the low temperature, the central fatigue contribute to this thing, to contribute to muscle fatigue. And then something else is of course, oxygen. What if the oxygen is used up? Not enough oxygen causing the let me see. Yeah. What if there's no oxygen supply, and this hypothesis also cannot explain everything, because even though we control our muscle activity in a very low rate, make sure the muscle has fully oxygen supply, there's still fatigue happen. So, and then why it happen? Another example is a glycogen. It uses up a glycogen. Once this has experimental support, because if the muscle start with full glycogen, it does take longer for the muscle to get fatigue. And for example, you have very good rest, it does have take longer to get fatigue, but still cannot explain everything, because when the muscle get fatigue, people have done the experiment that there's still glycogen left over in the muscle. It's not fully used up. And then probably the most promising target is RYR. We have mentioned this. RYR is on the top. This is the cap. This is the cap of the calcium, the calcium reservoir, the sarcoplasmic reticulum, the calcium reservoir. And then during the muscle contraction, it's not like this always open or always closed. It's kind of constantly open, close, open, close, open, close, to generate a calcium fluctuation. And this calcium fluctuation is very important for the muscle contraction. It cannot go always high or always low. Either the muscle cannot contract. And then think about this one. This is a small, tiny protein. Open, close, open, close. Eventually, it's worn out or torn torn out. It's the protein just denatured, it has no more function. When the protein denatures, calcium leakage out. When it leakage out, it's at the beginning it's like this and then fluctuation, nice muscle, and then when leakage, the fluctuation become worse and worse, and then no fluctuation, and then the muscle strength become weaker and weaker and weaker. And so, and then why you can gradually recover, because the muscle can produce more RVR, a new RVR to replace an older one to cap this reticulum, the sarcoplasmic reticulum. And here, this probably the most promising explanation of the muscle contraction, of the muscle fatigue. And lastly, let's talk about muscle regeneration. We have mentioned regeneration in almost all the tissues. What is the lowest regeneration? Is the cartilage. Cartilage basically has no regeneration at all. And slightly better than cartilage, the ligament, a little. And then what's better than ligament is the tendon. Tendon has called tendon stem cell progenitor cells. These cells may promote to ligand regeneration, which make it a little bit better than ligaments. And bone is much better than tendon or ligaments or cartilage. And what's the best? Muscle. Muscle regeneration is superior regeneration. Is one of the this is a muscle, and on the surface of muscle here, this are called satellite. Satellite cells. Satellite cells are stem cells. They are stem cells. Whenever the muscle is damaged, like say something here, the satellite cells will produce into differentiate into myoblast. And myoblast joined together called myotube. And myotube become a muscle, become a new muscle fiber. A new muscle fiber, or kind of, yes. So that's the and also in the muscle you also have multiple nuclei. Multiple nuclei, they can migrate. Whenever see this as something wrong, all the nuclei come into this position, and to make mRNA, to make more proteins to fix the damage. And of course, you have a lot of blood vessels running through the muscle. Also provide a high property of high regeneration. And then this damage. And what can most activate the satellite cells? Damage. Damage trigger the satellite cells. So damage, repair. And when you have a muscle damage and repair, in some cases, the muscle becomes stronger. And this pattern is used for training. Most of the training have a lot of a cycle of damage and repair, damage and repair. Through this one, you can achieve multiple things. One thing is that thicker myofibril and more myofibril, and this together causing a thicker muscle fiber. However, can we get more muscle fiber? The answer is no or hard. There's some evidence showing that maybe people with proper training can get more muscle fiber, but overall, it's extremely hard to get more muscle fiber. And there is another type of a training, not really put the muscle fiber. Some of this muscle fiber. One training can ignore last week. So in this case, in the proper training here, at the beginning, this is a muscle fiber. Muscle fiber. Through training, you get more. I'm just doing dramatic here. You have thicker and more myofibril through the training, and the whole muscle fiber becomes thicker. There's another way. You can also increase the muscle fiber without increase the fibril, the myofibril. This is only increase the content of glycogen, and this is content glycogen making muscles look fuller. This is more used for muscle builder, and this is more used for the athletes of strength training. So I think that's it. That's it for the muscle. See you next time.